For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started.
All right, looks like we are live, guys, and we are ready to go. I'm excited for this discussion. I'll say 2021 has been a blast so far, very eventful. We've had a lot of shows, interviews, discussions, debates. So I'm glad to be having these important discussions. Tonight, specifically, I'm having a discussion with a rational empiricist on ancestry and nested hierarchies. It's going to be similar to the discussion last week with Luca and myself on independent origins. We'll start off with some introductions, and then that'll lead into an equally timed, cordial discussion on the issues and the topics at hand. Real quick, before I hand it over to Rational Empiricist, for him to kind of break the ice and give an introduction of himself as to who he is. For example, I'm sure he's no stranger. Everybody's going to recognize him from the community. But a couple announcements. So on the 4th of February, we're going to have Michael Ord on to discuss the flood. So I'm excited for that. Anybody who knows Mike Ord will know why we're so excited for that. He's one of the top young earth creation flood researchers. So we're going to have a great presentation from him and some questions and answers. We've also got some other debates planned, possibly one next week with Kent Hovind. I'm just waiting for a final confirmation, but point of the story, lots to look forward to. Enough for me, though. Rational Empiricist, thanks for being here. You are on mute, so when you speak, my good man, make sure you unmute yourself. Why don't you give a brief introduction as to who you are, and thank you for being here.
Yeah, my pleasure. So I go by Rational Empiricist. I am interested in things like evolution, astronomy, and I generally like to learn subjects. I've been studying these kinds of subjects for a while now, probably my entire career in school, if you could call it that. But yeah, that's pretty much all you need to know about me. That I'm curious, I like to learn, and I like to have discussions with people like you, Stanley.
Awesome. I appreciate that. And thank you to the audience. We got some good people in the chat. This is going to be a lot of fun. You can, I'll be periodically looking at the chat, so you can shoot some questions our way, and I'll save as many as I can if we have a chance. Yeah. Argatha said, because I noticed that too, Rational Empiricist, in our pre-show, your audio was perfect. But right there, it was like, it's like something quickly switched where you kind of sounded staticky. I'm not sure if you're using a headset or something, you might have nicked it or something. I don't know, but it suddenly went fuzzy there. Do you know what it could be possibly?
Is it working now?
Let's see. Do it again. Do a testing.
Testing.
It looks like it's improved to me. Yes. Yeah. So I mean, whatever the issue was seems to, the roboting we heard there for a few seconds.
Oh, sorry about that.
Oh, no, it's not your fault. It seemed to happen abruptly too. And you sounded perfect in the pre-show. So whatever you did, it seems fixed. So what we're going to do, guys, we're going to keep this easy going. We've both got some slides and some brief presentations just to kind of go over the position so we can understand each other and represent us, each other soundly and accurately. So we're going to let a rational empiricist take as much time as he needs. No timer, just, yeah, whatever you need to present. Go ahead. I can share your screen as well. And when you're done, I'll do the same, take equal time, and then we'll get into a discussion. So whenever you're ready.
All right. Let's see how this works. All right. How's it doing?
Looks good. I'll make it full screen so the audience can see better. Let's see, full screen. And boom, good to go.
All right, let's get this started. So the question I want to pose tonight are, are nested hierarchies with common descent or common
Actually,
design.
I should stop you before you continue. It looks like that roboting is back, which you
Uh-oh.
don't typically have. Do you think it could be? Are you using a headset or the computer mic? Do you know what it?
Okay, does it sound better?
Now it does now, and it did earlier too. And then once you,
Hmm.
so hopefully as you continue, are you using the same mic or headset or whatnot that you typically use in the discussions?
Yeah, I'm just using my Chromebook. I don't know why it's doing this. I just keep muting and unmuting, so I guess I'll just have to do that periodically.
Okay. Okay. Yeah. Go ahead. You're
All
good.
right. So this is the question tonight. Now, obviously, I think the answer is common descent. Um, Now, first, I want to talk about what exactly relatedness means. Now, saying, for instance, that you are related to your cousins and your siblings, and that you're more closely related to them than you are to George Washington, for instance, simply means that you're stating that you share a more recent common ancestor with your sister cousins, and
Oh,
your siblings.
I'm sorry. I'm sorry because I want because you got a good presentation, good slides here. Maybe let's try because if it just comes out you roboting the whole time, people might not get the full
Yeah.
experience of your presentation. Maybe we should try because you sounded good in the pre-show. Did you want to leave the stream, kind of refresh your browser, and then make your way back in, or?
Yeah, I guess we can try that.
Because you said yeah, okay. So we'll try that, guys, just so we get all of his arguments that he's using. Let's make sure that everybody can hear it. So let's see how the chat's going. We got Bill Morgan in the house. Good to see you, brother Bill. Always a pleasure. Luca, good discussion last week. I appreciated that. Honesty Angel, Caustic Soda, Redefine Living. Lots of good people in the chat. So this is going to be a good discussion. I'm looking forward to it. So we just want to make sure that all the possible audio problems are worked out in order that we can hear rational empiricists arguments and model here. So why don't we do another audio check here and
All right.
see if it's
How do I
improved.
sound? Uh-oh.
Yeah, you
Hearing
sound good. I mean, you sound good now. It seems like when
Huh.
so you sound good now, but once you get going into the presentation is typically when we saw the issue. So maybe
Maybe I can try sharing my screen.
okay, yeah, let's do it.
Let's try that. Sorry, we may have some difficulties here.
You sound good now, so unless it's just when you share your screen, then it takes away too much bandwidth or something, but let's try that. So share your screen and hopefully, here we go. Because it must be just from your end because people in the chat are saying, guys, let me know how my audio is if I'm roboting out or anything like that. Okay, yeah, maybe go through that slide. I would start from the beginning.
All right.
I
don't know why it's doing this. All right, so I guess to go over it again. So by relatedness, I mean that you are more closely related to your siblings than you are to your cousins, and your siblings, and your cousins are more closely related to each other than all of you are to George Washington, for instance.
So I think I might have figured out what the problem is now. So
Okay.
which means if this is the case, it might just be when you have your screen shared. So unshare your screen and then do it and read a slide without the screen share. If the problem's fixed that way, then we'll just have to bear through your presentation with a little bit of crackling. Yeah, try and do it without the
All right, how about now?
Yeah, so right now it sounds good. So maybe read through your first slide without sharing your screen. Let's see if it solves the problem. If it does, then it's clearly whenever you share your screen, which means You could still do your presentation with your slideshow. We'll just know, hey, it's going to be a little crackly during your presentation, but afterwards, when we discuss the issues, then we won't really have to worry because you won't screen share as much. So maybe do this. Read off your first slide right now so we can test it, and let's see if it fixed the crackles or the roboting. Go ahead, start now.
All right. So you share a more recent common ancestor with your siblings than you do with your cousins. That common ancestor is, of course, your parents, specifically your mother. And you, your cousins, and your siblings share a more recent common ancestor with each other, your grandmother or grandparents, than all of you do with, say, your with George Washington, which great, 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 etc. grandmother, for instance. Concept is well, first of all, how's it going so far?
It seems to be in and out. So I mean, we might just have to deal with it. I'm not too sure what it could be going on your end. Is there another? So you're using your Chromebook, which you say you typically use. Is there another? Is there a laptop or anything you want to try out with? I mean, we can do our best to just deal with it, but at the same time, I want people to be able to understand what you're saying and your arguments without crackling out, every few seconds. Are you just using the Chromebook microphone, or do you have anything plugged in? Like, are you using a headset or anything?
I'm just using my my Chromebook. Hold on, let's try. I'm gonna unplug my mouse here. All right, how's it doing now?
Yeah, right now it sounds good because usually it happens, like it'll start kicking in every few seconds. Unplug your mouse. I guess anything that you can do to improve it, and we'll just hopefully it kind of resolves on its own. But if you're doing the same thing you typically do, then it's tough to say what it is exactly. So go ahead. I apologize, but we'll deal with it. Hopefully it resolves on its own. So go ahead, man. Go ahead.
All right. Can you see my screen first of all?
Yes. Yeah, you're all good.
All right. So with that example, the same concept can be applied. The concept of shared history can be applied to living populations, just using common ancestral species instead of individuals and ancestors. So how do we determine relatedness? Well, we simply find a pattern of more and more recently appearing traits within various groups, or taxa, as they're called. Now I'm going to go over the simplified steps in building phylogenies. So the first step is to compare all or most of the characters of the taxa under study using what's called a character matrix or data matrix. It's also called an array in computer programming. You look for the uniquely shared traits in the taxa under question using a computer algorithm because, of course, there are many, many different characters that you can look at for various species. And, of course, the number of species you're looking at can vary as well. Next, you use those uniquely shared traits, also known as synapomorphies, to group organisms into what are called monophyletic clades. So these are clades that include a common ancestor and all of its, no matter how different those descendants might be. Then we utilize different algorithms to determine which of the competing trees that we came to is most likely given the data that we have. And then finally, we make and refine the trees based on new evidence. And those are the basic steps. So then going into the steps more deeply, first we construct a character matrix like you can see here. We basically list the states of the characters for different species, the taxa, on one side, and on the other side with traits and their presence or absence. This can also be listed as various states, so it doesn't have to be binary, just 0 or 1. It can also be 0, 1, 2, free, etc., etc. So we list out all those characters for the taxa. This can be tens to hundreds of characters in total. These characters can also be based on morphology and or molecular sequences, typically DNA or protein sequences, and they often corroborate each other. So then the second step, to group based on synapomorphies. So you can see here, there are certain synapomorphies, certain uniquely shared traits that the taxa share. And I've basically labeled these A, B, C, D, etc. What is synapomorphic versus what is simplesiomorphic? Now, the difference here between synapomorphic and simplesiomorphic is that a synapomorphic trait is shared by the taxa in question, but is derived. So basically, it wasn't found beyond that group. It defines that group. So, for example, if we're looking at vertebrates and the trait vertebrae, comparing vertebrates to invertebrates like sponges, for instance, vertebrae would be a synapomorphic trait because only vertebrates have it. But if we're just comparing all vertebrates, vertebrae would be a simplesiomorphic trait because it is shared by them but is ancestral. So I just showed that there. The next step is to construct all possible trees, such as this one right here. This is just a consensus tree, but you can make many, many different trees based on the different traits. And there are many, many different trees for even a few taxa, and this number increases exponentially as the number of taxa increases. So, for example, there are more than 10 to the 80th power possible trees for over 20 taxa or so. That is, in fact, more possible trees than there are atoms in the visible universe. This does not mean, however, that all possible trees are equally likely, and so we need algorithms to assess which of them is most likely given the data. So then we get to the fourth step, which is to assess competing trees. In modern phylogenetic analyses, this is often done using one of two principles that are based on different assumptions. The first to be developed was parsimony, and this is the tree with the least amount of steps or character changes involved is the most likely. And this is mostly used for morphological characters nowadays. It can also be used for molecular characters, but is less used nowadays than the other methods, such as those based on probability, which means the tree with the greatest likelihood of individual steps given different models of DNA substitution, different mutations, basically. The tree with the greatest likelihood of those steps is the best given the data. And this includes methods such as maximum likelihood and Bayesian inference, which are similar but subtly different. They're the most common methods used today, but they are computationally intense. So sometimes it's better to use one method than the other. Methods involving overall similarity, such as in molecular phylogenies, aren't utilized much nowadays, but some methods, such as what's called neighbor joining, are used when certain conditions are met. So they can be used for a variety of different species when there are many different species taxa in question, and they can generate a tree very quickly. But that tree is often just an approximation of these other methods. And then the final step here is to refine the likeliest tree by making predictions. So, for example, we predict using evolutionary phylogenetics that So a fossil record will contain certain transitional forms that reflect what the hypothesized common ancestors may have looked like. Ancestry is basically concluded from phylogenetics. It isn't assumed. It is true that evolutionary phylogenetics is based on evolution, based on other evidence. And it is true that in order to root the tree, to basically have one species as the stem group, you need to use outgroups or really, really old fossils to figure out what exactly the transition is in terms of each character. So you need to figure out what's called the character polarity, going from ancestral to derived traits. And so we can make predictions about past taxa based on extant groups' character distributions. So for example, this is not what we would expect under evolutionary phylogenetics. Nothing like a crocoduck. So to conclude, basically, or to sum it up, phylogenetics makes predictions about the data for extinct and extant species that have all been verified, at least according to my position. Common design, on the other hand, doesn't make accurate predictions because it is not based on the assumption of uniquely shared traits showing a history, basically. Uniquely shared traits are a pattern that reflects an evident history. A nested hierarchy of derived traits within more and more inclusive groups, so going from vertebrates to amniotes to mammals to rodents, etc., doesn't suggest common design for the same reason that the traits of cars don't suggest that they have a history. There is no reason to predict a nested hierarchy in living things due to common design because common design does not produce a pattern of shared history, essentially. So then my final slide would be step 6, ask questions. So standing for truth, basic questions to kind of focus on. How would you predict the pattern of shared history in living things if such a pattern is deceptive, meaning that it looks like we're related, but we're not actually related? How do you explain transitional fossils in the geologic record, which you have to contend with? And finally, how do you determine which models of common design are more accurate? In other words, how do we predict what the creator should or should not have done based on the traits we see? And that's about it. So hope that presentation kind of summed up my position.
Awesome. I appreciate it. I appreciate the slides and the visuals. Great opening, empiricist. So I appreciate that. It looks like, hopefully this is the case. It looks like when I unmute myself, then your audio is fixed, as in there's no static or roboting going on. I don't know why that would fix it, but let's hope it stays that way. I'll just make sure that I'm, I guess, unmuted the whole time.
Yeah.
Your Chromebook, by the way, because I think this is going to be a good discussion, and your opening was good, so it's a lot of good questions we're going to touch on. I want to make sure audio is as good as can be. Is your Chromebook, is it fully charged? Do you have it plugged in? Like, is there anything you can do to also make? Is there a low battery on it? Anything like that?
No, it's pretty charged actually. I mean, I can plug it in, but it's close to fully charged, so I don't know exactly what's going on.
And then you just use the built-in mic, I'm guessing, right?
Yeah.
Oh, okay, so you don't use a headset or anything like that.
No. Yeah.
Okay. Well then, okay. It looks like it's fixed for now, so hopefully we'll keep it that way. And great. I'll go into a brief opening. I didn't time you, so I'll just try and go as equally timed as possible, and then we'll get into those questions and a free-flowing dialogue. So real quick before I go into an opening presentation, I saw a super chat come in a little while ago. Here we go. Thank you so much. George Bond in the house says he's just listening and eating and drinking. Sounds like a party, George. George is on vacation, much deserved, well-earned vacation. paid for by Team Standing for Truth. No, I'm just kidding. So anyways, thank you for the super chat. And here we go. I'll mute you real quick, Empiricist, till I'm done. And
yeah,
let's get this party started. So before I share my screen, I will point out before I forget, watching his opening, right off the bat, I want to make a couple points. Phylogenetic systematics assumes right off the bat that all DNA differences are the result of mutations over time. They do not consider the front-loaded DNA diversity hypothesis. Okay, they also assume ancestry first. And I've got some direct quotes from the evolutionary community themselves demonstrating this. They also don't question the existence of the tree. Okay, that is assumed right off the bat. The traits they are looking at are all assumed to be the result of descent with modification. For example, the shared derived synapomorphies that he talks about focuses on the aspect of the word derive, okay, as if common ancestry itself is a fact. And we're obviously going to get more into that in the discussion portion, but I wanted to point that out while it was fresh in my mind. Let me share screen here, guys. And okay, I am going to make this full screen from current slide. Okay, so obviously since this is my channel, I'm no stranger to my own channel, but I'm standing for truth. I've written numerous books on this topic, the topic of ancestry. Your two books, Why Human Evolution is False and Universal or Separate Ancestry, are more basic for the layman, and then the other two books are more technical rebuttals to some of the best arguments from the critics. So definitely check those out if you're interested. Also, I've co-authored numerous books here with Ramat, which can all be found on our website, Creationist Clothing. So let's get right into this. This is going to be a lot of fun, one of my favorite topics. Andrew today, as you've seen, he's arguing for universal common ancestry, okay? Well, I am arguing for independent origins. Now, essentially, Andrew can argue from homology, which can be applied to homologous patterns in anatomy. You can look to homology in embryology, but also the genetic realm, okay? Similarities exist in the biological world. Nobody that I know would argue against this fact. Now, Andrew may also argue for universal common ancestry by looking to the rare so-called transitional forms, okay, such as Tiktaalik, or the mammal-like reptiles, or Archaeopteryx. There's a number of them. As in those types of animals, let's say Tiktaalik, that appear to blend the features of different varieties of animals, okay? And most importantly, as my interlocutor here did in his opening, he can look to nested hierarchical patterns, right? Those groups within groups patterns that exist all throughout the biological world. Okay, the fact that humans are more similar, for example here, more similar to the great apes than they are to the old world monkeys. And essentially both humans and the old world monkeys are more similar to each other than either are to, say, a dog or a fish. This is the structure of life. We can classify and categorize creatures in these groups within groups patterns that evolutionists assume are the result of descent with modification and ultimately universal common ancestry, okay? Humans, as you can see here, are grouped with primates, which is basically because we share much more in terms of anatomy, morphology, physiology, and genetics with them than we do with fish. And yet, we are still grouped with fish because we share more with fish than we do with insects such as spiders and butterflies, okay? Proponents of evolutionists, they will say that the amount or degree of similarity between creatures is actually a reflection of time since their last common ancestor. We can even look to the genetic sequences. We can look to nuclear DNA, mitochondrial DNA, and what we find are nested hierarchical patterns. Do all these lines of evidence, this is the question, do all these lines of evidence indicate a process of descent with modification and universal common ancestry, or design and creation? Okay, now before I get a little more deeply into it, I want to point out that using these types of arguments, homology, transitional forms, nested hierarchical patterns, to support universal common ancestry would be like looking to the sky, okay, and pointing out the fact that it's blue as a way to argue for evolution. This would be ridiculous, since other models can also explain why the sky is blue and why the earth is round, let's say. The blueness of the sky or the roundness of the earth can be both explained by evolution or creation. Okay, so when it comes to homology, human designers build in homologous patterns. This is what we see around the globe. We also see homologous patterns at the blueprint level. Oftentimes, guys, the earliest stages of different types of modes of transportation, let's say sedans, SUVs, vans, they all look very similar, but it's not until the final product that we see the most differences. Okay, so as you can see here, humans build in homologous patterns. Transitional forms, okay. Humans also build, not to demonstrate the creation model, they do it for functional purposes. They build vehicles that seem to blend the features of multiple types of vehicles. One such vehicle that comes to mind here that I've got in the picture is an amphibious assault vehicle, which happens to blend the features of both a land vehicle and a vehicle built for the sea. This is all good design. It's also good forward thinker. That points us back to the forward thinker. Okay, this forward thinking that we see. Human engineers also build in nested hierarchical patterns. These types of groups within groups patterns are found everywhere in the design world. In other words, we can find an echo of the overall pattern of life in the design realm. We can see hierarchical patterns at the visible level and the blueprint level. Now, evolutionary responses to these observations are typically very unsophisticated and also based on misrepresentations of the creation model, okay? A typical response I hear is, cars don't reproduce. We've all heard it. For one, adding reproduction adds complexity and also makes the problem worse for evolution. And two, this is a strong misrepresentation of our model of independent origins, which I am happy to get into in more detail during our discussion. I also want to point out that man is producing these vehicles, okay? These modes of transportation. They're creating them for a purpose and for a function, an intent to move somebody from one place to another. God produced life. He created life. And he has given his creation the ability to reproduce and pass on their genes. This is amazing. This is beyond anything man has ever been able to engineer. Cars, for example, are all made out of natural material, iron, for example, okay? This doesn't mean cars made themselves. Humans, animals, plants are all made out of natural occurring elements from the earth. Humans, animals, and plants did not make themselves, okay, in the same way that cars and other modes of transportation did not make themselves. Somebody made them. And from the natural occurring materials found in the earth, man has dug up these materials. They've melted them down and used the iron and the steel. Okay, I'm simplifying this, of course, for the point at hand, but they've used metals to make cars and modes of transportation. Man can do this. God can do this. But these materials themselves cannot do this. This is the point of the design model. And we can get a sense for how God may have created the biological world based on the way we as humans design things. We are made in the image of God, of course. Anyways, I'll end it with this, okay? Because the data strongly indicates that these hierarchies are here by design. And yes, we can classify life into groups and categories based on anatomy, morphology, and genetics, for example. And by definition, humans can be classified and grouped within these man-made classification systems. To say that humans should be equally apart or equally distant from all forms of life, okay, as compared to more similar to some creatures, such as the great apes, and less similar to other creatures such as fish and bacteria, is just a horrible argument. It's not sophisticated, and it considers very little. Evolutionists such as Aron Ra and their straw man phylogeny challenge seem to be proposing that man was created without DNA, RNA, and proteins that make up life and ultimately code for the anatomy and the morphology of living creatures. This type of design wouldn't make any sense, guys. It would also hinder scientific advancement in terms of study and research. Brother Salvador Cordova points out the fact that these similarities in structure, in genetics, for example, in physiology, anatomy, morphology, are important when it comes to healthcare and various medical fields. Since, of course, similarities in DNA help with cures, they help with medicine. Okay. Now, the differentiating lines of evidence, this is the last thing I'll say, comes down to function. Okay. And there are some inconsistencies and lack of uniqueness in the phylogenetic tree, which we will get into during the discussion portion, convergent evolution, for example. A lot here on that, which we'll discuss, as well as incomplete lineage sorting, major problem for the evolutionary phylogenetic tree. Taxonomically restricted genes, another major problem. Epigenetics. So the last thing I'll say is, when it comes down to the differentiating lines of evidence, it comes down to function. Okay, this is not a long process of evolutionary advancement, evolutionary history. These hierarchies are there by design and for functional purposes. In past debates I have looked to, with no answer or real rebuttal by the critic, human-specific genes, multifunctional and multi-purpose genes, differential gene expression, unique transcription factors, non-coding DNA, and it's amazing. It's many amazing functions not predicted by evolutionists. The FOXP2 gene, signs and lines, long non-coding RNAs, genomic networks, and more, okay? All of this demonstrates independent origins and refutes universal common ancestry, and essentially demonstrates that these nested hierarchies are there by design and not by consent. So I will end with that, and I'm going to hand it over to a rational empiricist. Take your time, address whatever you'd like to. I'll let you kind of kick off the conversation and take it any direction you'd like it to.
Yeah, sure. I didn't know if you want me to like give a rebuttal, or if you want me to just kickstart it with some questions I have for you, or how would you like to do it?
However you'd like to. You can start off if you wanted to rebut a couple points, and then finalize that rebuttal with a question, and then we can go from there. That might be a good way to start if you'd like to.
Okay. So I guess one thing I would say is that it is possible to build a what looks like a nested hierarchy for almost any kind of object. Take the planets, for instance. Now, obviously, we may disagree on whether they were designed or not, but you can put the 9 planets and other exoplanets possibly into a nested hierarchy. You could say the gas giants fall into one group, and the terrestrial planets fall into another group, and then you can differentiate them. Now, obviously, this isn't as complex or branching as proposed evolutionary models, but I think what this demonstrates, or at least demonstrates to me, is that there's a difference between a nested hierarchy that is based on subjective it's based on certain characters being made more important before others, while in evolutionary model, if you essentially let the data speak for itself, now some people might say we're interpreting the data differently, but if you let the traits and characters arrange themselves, essentially, then you naturally get this nested hierarchy. This isn't the case for designed objects such as cars, for instance. You can arrange them to make them look like a nested hierarchy, but it doesn't naturally flow from the data there. And this is the other thing I want to mention. which was that even though we have a nested hierarchy, the nested hierarchy isn't perfect, basically. That there are certain methods that are needed to find how traits compare with one another such that we get a consensus nested hierarchy, I guess I should say. Whereas if you were to take designed objects, that means that their traits have to relate to their function. So I guess a question I might ask would be, for instance, we see lungs, mammalian type lungs in, say, whales, which are adapted to live in the sea. Now, how do leftovers like that, if I were to call them leftovers, how do those kinds of traits fit into a common design model when certain traits such as that are not related to function necessarily? So I guess I'll leave it at that.
Okay, good question and good points. Let me address a couple of the points and then we'll get to the question, just so we don't leave anything hanging. I will point out before I start that Linnaeus, the father of taxonomy, was a young earth creationist, and he based his classification scheme on what? He based it on function, right? Kingdom, phylum, class, order, family, genus, species. Okay. And he was a young earth creationist. So creationists have always understood that these patterns, these nested hierarchies we see should then be there by design. Now, obviously, as technology improves and our understanding of the design world increases, we are now better at recognizing the characteristics of design. When it comes to vehicles, when it comes to modes of transportation, okay, that's what I focused on. And you can also see hierarchies in computer code with the functions, sub-functions, and so on and so forth. But when it comes to modes of transportation, okay, this is not being done on purpose. Okay, these engineers are not saying, hey, let's build these vehicles in these types of patterns in order to demonstrate the design model. No, it happens naturally. Okay, for example, we can look to, let's say a sedan. Okay, a sedan has a tremendous amount of similarities with other sedans. Okay, now we can bring in something a lot different than a sedan, let's say a semi-tractor trailer. Okay, so we know it's a lot different than a sedan or a car, but these two modes of transportation are similar enough to each other that it still sets them apart from, let's say, airplanes or boats. You know, this is the groups within groups patterns that emerges when we look at all these different modes of transportation and vehicles that humans have designed. This is an empirical observation, essentially. So our model would say this, and this is why I focus on these patterns in the design world, is because the Bible states that God created us, created man in his image. Okay, that means we should be able to get a sense. This is where the predictions come into play. I noticed this was one of your questions. Therefore, we should be able to get a sense for how God designed the biological world by how we design things. And yet it just so turns out that we build and we create in these nested hierarchical patterns, okay, all throughout the design world. Now, here's the thing. You may find some inconsistencies. Okay, ultimately, the pattern's there. Okay, both models predict a hierarchical pattern. But when it comes to the evolutionary model and the phylogenetic tree, you are finding inconsistencies, which we can get into a little bit later because I know you had a question there, such as convergent evolution, incomplete lineage sorting, taxonomically restricted genes. For example, orphan genes have no consistent hierarchy, none at all. These mosaics that we find in the biological world, very inconsistent in terms of taxonomy and cladistics. But I'll say this too, because you brought up a lot of points there. When it comes to cladistics, okay, and classification systems, important to notice that I'm going to share a screen here real quick. So I want to show a source, and then we'll focus on your question here.
Sure.
Just 1 second. So let's see. So, and what's good is based on your question, you seem to understand the fact that, hey, listen, descent with modification has a long evolutionary history behind it. Okay, that's why on a genetic level, what we should predict to find are evolutionary leftovers, genomic fossils, a reflection of this long history of evolutionary advancement, for example. While according to our model, we would predict function, very little evolutionary leftovers, very little genomic fossils. But here, so cladistics, okay? Cladograms only demonstrate a nested hierarchy of biological characters. They tell us nothing essentially about what produced the pattern, right? So looking at these cladograms, but then coming to the conclusion that this is all due to fish to fisherman evolution, descent with modification, universal common ancestry, okay? That's an inference of the data. That's why we're here today to obviously discuss what produced the pattern itself. Right here, where is it? Cladistics enables us to gain a picture of the nested hierarchy of life, and that's what we're doing here. But the method itself tells us nothing about what produced that pattern. Since evolution wasn't needed either to discover or understand the pattern, it's not evidence for evolution. And I want to show here from a secular source. They assume the, where is it? I had a source here where they assume the tree before they, let's see if I can find it. Right here. The key assumption made when constructing a phylogenetic tree, here's the paper here, from a set of sequences is that they are all derived from a single ancestral sequence. For example, they are homologous. And same here. So right away, they assume the tree, they're doing the phylogenetic analysis, and they are coming to the best inference that's possible, okay, in light of the data. But they're not necessarily questioning the bigger picture, or else you'd see them in their quotes, in their technical paper, pointing that out. Anyways, I know I said a lot there because you brought up a lot of good points. Comment on any of that, and then let's go right into your question, or we can get right into your question right now, of course.
Right. Yeah. I guess before we get into my question, one thing I might point out is that it is true that we're assuming some kind of shared history, of evolutionary history, of ancestry, basically, in these models. Like, but the point is that unless we assume those kinds of histories, you don't get a nested hierarchy, basically. So if you assume, for example, if you say, for instance, that cars have a history, let's just give a hypothetical example here and say that cars evolve through genealogies, basically. What you would find is that given the traits, that you wouldn't get a nested hierarchy, you wouldn't get these kinds of relationships. Because we know that cars are designed. So we know that they do not have a history. Now, what we can see in living organisms is the pattern that reflects a history. So I just wanted to make that point real quick. And so I guess my question would be, if you're comparing, say, for instance, because there's always going to be character conflicts. I think we both agree on that if you take a tree, there are going to be conflicting characters. Now, what you find is that if you have common design, basically the overlap, the character conflict should be a lot more severe, basically, than the broad models of evolutionary history. And we find that history goes back even further. So I don't want to go too much into that, but what are your thoughts on that?
Good points, good question. I would say that naturally, Okay, when we're looking at, I mean, just for the audience sake, let's show some diagrams of what we're kind of discussing here. Okay, so
Okay.
go right up to here. So naturally, just standing back, okay, when we see these natural nested hierarchical patterns. Now, I will say, I believe you're demonstrating a bit of circular reasoning when it comes to taxonomy and cladistics, okay? Because I find evolutionists, they do hone in on all the similarities. But as I was pointing out in my opening, we can touch on the taxonomically restricted genes, the incomplete lineage sorting, the convergent evolution. There's a lot, okay? There's a lot of these inconsistencies, but they're never questioning the bigger picture. So they're still force fitting it into the model because it all goes back to the fact that they assume the tree exists, as in the universal tree. Because we believe in trees, essentially. We just believe in independent origins, right? You'd have a separate tree for humans, because God created two humans, a separate tree for, say, the cat kind or the canid kind, for example, which we can get into. But I want to point this out, for the audience, for yourself. When you just stand back, okay, even before we had genetic sequencing, you could look at a human, a chimpanzee, a mouse, a lizard, a salamander, and a fish. Right off the bat, you can stand back and just based on anatomy and morphology alone, okay, you could line this up as a human being more similar to a chimpanzee than a human is to a fish. Even the human and a mouse would be more similar to each other than the human and the lizard. You could just see this. And even if you want to go just into apes, old world monkeys, new world monkeys, right off the bat, based on anatomy and morphology, you'd conclude that apes and humans are more similar to each other than they are to the lemurs. This is all just expected, predicted when it comes to both models. The evolutionists will say humans and chimpanzees share a most recent common ancestor. Okay, humans and chimps split, they say, about 6 million years ago from a common ancestor. So they have a more recent common ancestor than, say, the human and the mouse, right? This is all a reflection of time, according to the evolutionists. And here's your synapomorphies, right? Shared derived features, shared derived traits and characteristics, okay? So they're assuming the derived, right? They're assuming that this has all been through a process of descent with modification. So once the genetic data came out, then yes, overall, we see a hierarchy there as well. And that's why I point out, even with the modes of transportation, you're going to have more similarities with a sedan and a truck than between a sedan and, let's say, an airplane or a boat or a bicycle. These are all just natural. So that's why I think it comes down to function. But I do think the evolutionists downplay the significance of the inconsistencies. Okay, for example, convergent evolution is a huge problem for evolution, okay? And we can let you comment on that. For example, convergent evolution, in evolutionary biology, convergent evolution is the process whereby organisms not closely related, right, not monophyletic, independently evolve similar traits as a result of having to adapt to similar environments or ecological niches. And then there's a few examples. You can see it in nature with flight, wings, on, let's say, insects, birds, pterosaurs, bats. These have all evolved independently, but they still serve the same function, and they're similar in structure. So all of this convergent evolution, okay, anytime convergent evolution is invoked, this is an admittance of a lack of uniqueness in the phylogenetic tree. Now, I'm not saying hierarchical patterns don't exist. I'm just saying there are some problems where now evolutionists have to say something as complex as a wing and flight had to evolve not once, not twice, but over a dozen times. And we can see the same thing with eyes. We can see the same thing in a number of characteristic structures and features. So for one, I guess, how would you explain convergent evolution empirically?
Right.
And two, why is this not essentially an inconsistency with the phylogenetic tree that you're saying is just so consistent and so expected given biological evolution over the design hypothesis? Go ahead, take as much time as you need.
Right. So if you could you pull up I saw a picture of a platypus there, for instance.
Yes, right there.
All right. Now I guess my point would be, even when you look at convergent structures, homoplasies they're called, what you find is that usually they still fit within the nested hierarchy, for instance. So for instance, I know that the bill of the platypus, for instance, is a different kind of material. I believe it's a different kind of keratin, basically, than birds' bills, for instance. The hair obviously is unique to mammals. The venom glands seem to be homologous with sweat glands in mammals, so they're not very much similar to let's see, the venom glands of reptiles, for instance. So they're not really it's not like you're taking one trait from reptiles and putting it onto this mammal. What you find is that they're usually modified versions of what was previously there, basically. And so I guess I would disagree with the fact that convergent evolution is a problem, because although convergent evolution can be hard to spot, when you look deep into it, basically, you find that these unique traits still fit this pattern. And the other point I would make is that it's not that there are inconsistencies, because like I said, there are going to be inconsistencies just by the fact that we don't have a perfect understanding of evolution, for instance. But what you would expect to see is that the level of inconsistency, this level of actual convergence, for instance, would be significantly lower in the evolutionary model of universal common descent versus common design. For instance, there can be a mix and match of characters basically between different vehicles. That is not what you would like. For instance, I showed the picture of a crocoduck, basically.
Right.
Now, obviously, no evolutionary biologists think that fossil forms in the past would look like that, basically, because we expect fossil forms, transitional forms, to follow this nested hierarchy, at least in the general outlines, because again, the fossil record is quite incomplete. But we would expect them to fill this nested hierarchy, not to combine traits from the tips of the tree, for instance. So you don't expect to find feathers on an alligator, for instance, like in the crocoduck. And that is more what I would argue we would expect to see in a common design model. Either the common designer, the creator, basically would use similar sequences, similar features for various functions like we see, or for similar functions like we see in human designs. But obviously that's not what we see. Like I brought up the lungs in whales, for instance, earlier. So that's what I would say based on what you said.
Okay, so yeah, so some good points there. I think it comes down to the fact, though, what we're looking at is a lot of inference, storytelling, maybe this, maybe that. The question is, can evolutionary processes explain the examples of convergent evolution that we see? Can they explain the examples of incomplete lineage sorting that we see? Can they explain the examples of orphan genes, taxonomically restricted genes? Because I've looked at a lot of papers, and there's never been empirical evidence demonstrating how these taxonomically restricted and highly essential genes to living organisms can come about through any number of mutations. Yeah, there's hypotheses, and they'll look to the genetic sequences of one type of creature and another type of creature, and they'll make guesses based on gene duplication, de novo gene synthesis, gene duplication followed by neo-functionalization, things like this. But a lot of it is based on assumptions, inference, and as I've shown earlier, the basic assumption that goes without question of the phylogenetic tree in the fact that there is a tree. There's no questioning the bigger picture. When it comes to all of these different traits on, say, this mosaic right here, because I pointed out in the beginning that we too predict mosaics, bill like a duck, and you're right, the materials, essentially, okay, the structure is the building blocks are slightly different than other creatures with similar structures, okay? Hair like a bear, poison like a snake. Point is, there's a mosaic of features here where, okay, it is a fact that taxonomists and those that work in these classification systematics, they've always had trouble classifying these types of mosaics. That's the point. Yes, because all of life is made of DNA, RNA, protein, okay, and the same type of structures, which are essentially coded by the information system that is DNA. Yes, you're going to be able to force fit it somewhere. And that's why I always point out that this whole phylogeny challenge, just show me this straight barrier, this straight line. Well, that's assuming God created humans, and then humans being equally distant from all other forms of life, right? Equally distant from chimpanzees, equally distant from fish. No, of course, God would create some creatures that are more similar to each other and other creatures that are less similar to each other. And this makes sense, too, when it comes to scientific advancement, especially in healthcare. We need to have these similarities and able to do these genetic tests in order to advance in terms of medical care, cures, diagnoses, things like this. This is good. This would be good design, good forward thinking. So my point is, it's the inconsistencies, because oftentimes I find the evolutionists are pointing to some inconsistencies in those natural hierarchies in the design world, okay? Well, the point is, overall, that's what you see, and some evolutionists can deny it all they want, but that's what natural hierarchy in the design world. In this, I mean, you can go right down to the material level and you're still going to see hierarchies, okay? You can look at the metals used, for example. And here's the thing, though. I am one person, and I can essentially write books, I can build a car, I can design a house, I can make a variety of different designs, just like the designer. So yes, you might have similar structures, because I know you're pointing to the lung systems of different creatures. Okay, as long as it's there for functional purposes, that's what we would predict, that these hierarchies are there for a reason. And that's what Linnaeus assumed: kingdom, phylum, class, order, family, genus, species. He is the father of taxonomy, and he assumed, he was a young earth creationist, he assumed that this classification system had everything to do with function and purpose and creation. But here's the most important aspect, because based on everything you're saying, right, because you keep saying that evolutionists expect some of these contradictions, that it's not all perfectly fit within that tree, but they didn't predict what we now know in terms of incomplete lineage sorting, where you can look to different genes, okay, and you can come up with totally different trees. For example, I'll go to this one and I'll yield for a bit. When you look incomplete lineage sorting, this occurs with similar genes between taxa. The type of gene or DNA sequence used also produces different results depending on what gene you're using. So not only is there incongruence observed across taxa, but also across gene types and gene families. For example, if you look to, there's a paper here I got right here. One of the first papers, this wasn't predicted, by the way, to expose this problem of DNA sequence, incomplete lineage sorting in the area of primate evolution was published in 2007. And what they found was in homologous sequences that they selected, okay, present in humans, chimpanzees, and gorillas, in which they trimmed out the gaps, insertions, and deletions, despite, okay, so what they did is they still optimized the data. And even in despite of that, a majority of the trees showed high levels of incongruence compared to the inferred model of human evolution. Okay, and then the researchers, you can look at the paper that I documented over here. There's a number of papers here. Okay, so yeah, the researchers are oftentimes in shock over these incongruent species trees. And yeah, they'll look to explanations and they'll have hypotheses, but that is the biggest point to consider is a lot of these inconsistencies, okay, in terms of the phylogenetic tree, they're not predicted. The Y chromosome dissimilarity between humans and chimps and gorilla, not predicted. You know, so that's the point, is if it comes down to testability, predictability, the evolutionists aren't really doing a good job. But here's the thing, it all goes back to this, and then you take as much time as you want. The fact that I showed directly from their own literature, their own quote, they assume the tree exists. They assume this is all a process of descent with modification. Therefore, they're just going to force fit the data. And just like with the platypus, they're going to fit it somewhere. Because if you have a circle that's just massive, okay, then obviously whatever trait you look at, whatever genetic sequence you look at, whatever structure you look at in terms of anatomy and morphology, it's going to fit somewhere in that circle when you make it as large as the evolutionists do in terms of taxonomy and classification systematics. So go ahead, take as much time as you need. I got to grab a bottle of water, but it's just 2 feet away. So I'll still hear what you're saying. Go ahead.
All right. So I guess one, I feel like I'm lagging a bit. Oh, there we go. So one thing, I guess a question that I might ask is, for instance, how would we, so let's say if I'm getting what you're saying correctly, the creator, the designer, for instance, created these forms, and we would get similarities from that. I guess, are you saying that the designer in this model created forms and these traits became similar due to random correlation? I'm like, can you clarify that?
Can you reiterate that question? Are they there for? Yeah, can you reiterate that real quick?
Yeah. So if we say, okay, we have the designer basically creating these different forms, creating these original kinds, for instance, and we see these homologies basically. Are you saying that those kinds basically converged due to happenstance, basically, that they converged because what I guess I'm saying is that if you expect them to converge due to function, then you would expect certain sequences to be, I guess, reused basically, for similar designs to be used for one function and different design for a different function. But that is not exactly what you find in life. For instance, you find function correlated with form of the structures, for instance, and you find structures that are not correlated with function. For instance, like you see the this is a common example, but it's the only one I can think of at this point. You can find the limb bones of all mammals, all tetrapods. They don't correlate with function, but they are all similar. Whereas if you look at convergences, for instance, you can see or not convergences. You can find one trait that is used in a variety of different ways, for different functions, and this is what we see in life. For instance, like spider silk, or
Real quick, real quick. No, I just want to stop you there, not for the debate purposes. How many tabs do you have open in your Chromebook? I want to see if we can just improve the audio just a little bit, just so.
Oh, I
can
just have
move you in
my
there.
slideshow.
Oh, because I know on the Chromebook it takes up a lot of memory.
Okay. Here, let me try and
especially
close
for
it.
long. But usually, if it's just an overload of tabs, I just had somebody recommend that, and just because
How
I
about
noticed
now?
I muted for a second when I was getting some water and I had some background noise,
Oh
and
yeah,
right away you started roboting out on me. You know what I mean? So,
yeah.
I want to limit background noise as much as possible. But yeah, if you can save memory and bandwidth by, I guess, not opening too many programs. But okay,
Yeah.
I apologize. Continue. Take as much time as you need, of course.
No, I was pretty much done basically. I guess to sum up my point, how do we differentiate between function and or random correlation and a history, basically, a shared history pattern, I guess, is it does that kind of make sense?
Yeah, I think it. I'll answer that, but I want to go over the bigger picture real quick. In the fact that right now, the best way to, and I'll answer everything you said, but just before I forget, I want to make this point. The bigger picture of all of this comes down to what is the best way to determine ancestry, the best way to make conclusions when it comes to ancestry. Okay, it comes down to genetics, because we're talking about traits. We're talking about shared derived synapomorphies, for example.
Right.
So traits are inherently genetic related. Okay, so it's genetics, it's traits that are inherited sperm and egg. So that's why down on the genetic level is a reflection of the morphology, of the anatomy, of the physiology. Okay, so if the majority of the genome, okay, is there for a purpose, is there for function, and you've heard numerous times from evolutionists such as Gutsy Gibbon, Dan, they say over and over again that the majority of the genome is junk based on genomic fossils, non-functional evolutionary leftovers as a reflection of this descent with modification. While we say no, these hierarchies which exist, okay, on the anatomical level, the morphological level, ultimately on the genetic level, we are going to see hierarchies by design as well, especially with a lot of these highly conserved mitochondrial genes, let's say the cytochrome C, okay, where you've got the function for it, but you can see a hierarchy, a pattern when it comes to humans, the great apes, down to the old world monkeys, the lemurs. Why is that pattern there? They'll say, why did God make the gene so different, the protein so different for so many different types of animals? Why didn't he just use one function, one gene, one structure ultimately for all your tetrapods, right? Well, so that's the bigger picture. We're going to make predictions on function. Hey, this DNA position is functional for this purpose, for this reason, or you knock out this gene and it's going to lead to this disease. Okay, these types of predictions that evolutionists typically don't make because they assume most of the genome is junk, reflecting descent with modification. But I'll point out this, when it comes to designers in the real world, when it comes to cars, millions and millions of cars here in North America have the steering wheels on the left side because that's the way they were built. That's the way they were designed. That's the way we like it. While if you were to go to, say, Britain, they've got millions and millions of cars designed with the steering wheel on the left side. What does that prove? Well, it proves that we've got purpose, function, design that are oftentimes a little bit dissimilar depending on where you go. It's just a variety of designs. We see that in the biological world, a variety of different designs when it comes to mammals, tetrapods. But you also got to consider that we look to epigenetics. Okay, there's there's millions and millions of switches in our genomes that are just ready and willing to be turned on or turned off via the environment. Okay, so oftentimes we'll see similar structures in organisms that evolutionists, based on form and structure and differences in terms of form and structure and function, they'll say this is convergent evolution. But they'll still conclude that these organisms share common ancestry somewhere down the line, right? Us on the independent origin side, when you have similar environments, plus you have these built-in mechanisms, these forward-thinking mechanisms, we've got these switches where you enter an environment. That's why we see rapid adaptation now. These creatures that aren't necessarily related, but they're in similar environments, okay? These genes are turned on, and we can see rapid epigenetic adaptation, which means you might have a similar adaptive episode, similar form, similar structure, but not exactly the same underlying. Because isn't that what you're saying, that we've got all these variety of forms and structure? Why not? Well, why wouldn't the designer just use, one tetrapod limb and boom, that's shared across all the different tetrapods? Is that essentially kind of what you're saying?
Right. I would say that if you look, for instance, at different tetrapod limbs, you're going to see that when you're comparing all these different organisms, it looks like it appears that they have a shared history, basically, that they have this that these set of characters have changed, basically, and you can see it on the fossil record, for instance. I guess to address one of your points, now obviously you would dispute that the fossil record shows change. But say granting that for a moment, that the fossil record, let's say there's some change involved, basically. You would expect to see, so for instance, if you assume that change happened, which I would argue is based on other lines of evidence, if you assume that change happened, that there's a history, a very deep history, then you would see this kind of branching pattern, basically, and these traits would follow that history. And I would argue that that's what you see in nature, basically. Now, for a common designer, I guess my original point would be, you see similar forms and in certain places, in different forms in different places. And this fits an evolutionary model. Now you would say if it's a common design model. Now I guess my original question would be, why exactly does the design pattern seem to fit a shared history pattern, basically? Because I guess diverting the conversation a little bit, because we've been talking about genetics and stuff like that, and I kind of wanted to mention fossils and morphology, if that's okay.
I would just point, just to conclude that part, is, like I said before, why isn't every single car on this planet built with the steering wheel on the left side or the right side? Differences, my point, in design that we see in the biological world is not evidence for common ancestry, especially if those differences are there for the predictive reasons coming from young earth creationists, which those reasons are, as you understand it, which is good: function, purpose, right? So as I pointed out, I'm one person. I can write a variety of different books. I can build a variety of different cars. I can, if I was handy enough, I could build a variety of different houses. Variety just points us to the infinite maker. But the point is, that's what we see in the biological world, because remember, our model suggests we were created in the image of God. I'll reiterate that. Therefore, if what we see in the biological world fits that what we see in the design world, okay, then that's an expectation, a prediction, a retrodiction that's met. Okay. And I would have to, I guess, disagree, because you're pointing out some cases where you believe that's not what's seen. I believe that's exactly what we see. And we can even see, for example, let's take humans, pigs, mice, and ducks. Okay. So they're all essentially land dwelling. Okay. And we would say, based on the independent origins model, we would say they are separate kinds. Okay. They've descended ultimately from separate creations.
Right.
But because they are all land dwelling, okay, because they still will have and share similarities in terms of anatomy, morphology, physiology, genetics, guess what we're going to find? Well, we're going to find nested hierarchies, even within creatures that are, by definition, according to evolutionary theory, distant on the tree, right? So that's the point, is what we're going to find as a rule in the design world is hierarchies. What we're going to find in the biological world is hierarchies. But you can point to some inconsistencies in my model. I can point to some inconsistencies in your model, right? As you've done, and as I have done in terms of incomplete lineage sorting, convergent evolution, and
Sure.
Taxonomically restricted genes. That's the point. But here's the thing. The exception to the rule should not overturn the rule. And the rule is a hierarchy. Both models predict the hierarchy. So I don't want to spend an entire debate. I'm not saying we are in this one, but I'm saying in general, when I go into a debate, I'm not going to focus entirely on homology and say, hey, this demonstrates a common designer, or look to some mosaics and say, hey, this demonstrates a common designer, or look to nested hierarchical patterns and say, hey, this demonstrates a common designer. I'll tell you why, because common ancestry can explain the same thing in the same way both models can explain why the sky is blue, why the earth is round. We got to look to differentiating lines of evidence. And that's why I truly believe that the best differentiating line of evidence is function, because the evolutionists themselves would admit that they assume a lot of non-functional evolutionary leftovers in terms of anatomy, structure, and form, because that's what you wanted to get into next, which is fine. They would look to vestigial organs, which is a reflection of that whole junk DNA paradigm, where you're looking at essentially junk organs, junk structures, right? So that's the rule. And we can pick at each other's models all day and say, well, what about this? What about this? It's not going to overturn the rule, though. We see a hierarchy on both sides, and I think both sides should be honest enough to at least admit that. And I'll yield, say whatever you'd like to, and then we can, I know you wanted to go to the fossils and morphology a little bit more, so go ahead.
Yeah, what I wanted to get into fossils basically was, I think one disagreement we're having is that, to me, this pattern of shared history goes back all the way basically to a universal common ancestor. Now, you would say, if I'm understanding your position correctly, that this set of shared history only goes back to certain kinds, and then they radiated off the arc, as I understand it. Now, if, for instance,
well, if I could, and I apologize,
okay.
and then continue as long as you want, because I just want to make my point absolutely clear. When it comes to the models, when it comes to your explanations for, say, convergent evolution, okay, or these similar structures, okay, that oftentimes you still find, even within tetrapods, you still find some dissimilarity in, say, a tetrapod limb, as you were pointing out. But here's the point. When it comes to the universal common ancestry model, the innovations, the body plans, the structures, the novelty that needs to be demonstrated, that's required if fish-to-fisherman evolution is true, that has to come about through natural selection, acting upon essentially random variation created through random mutations. And as you know, a primary argument from intelligent design advocates is the fact that natural selection, okay, even epigenetics and mutations, lack significant explanatory power in regards to the origin of these types of novelties, the origin of, say, phenotypic complexity, anatomical novelty, or even just abrupt fossil appearance that we're seeing in the fossil record. And this last thing I'll say, when it comes to the genetic level, well, now the evolutionary community, these evolutionary mechanisms like natural selection and mutations, they now have to explain how this four-dimensional genome that's nested, okay, it's nested and it has multiple overlapping codes. And these multiple overlapping codes are chock full of meta information. Now they got to explain how, on a genomic level, a molecular level, how this all came about. So, I just wanted to point out that I don't believe the explanations for the inconsistencies like orphan genes, incomplete lineage sorting, convergent evolution. I don't believe they are empirical. I don't believe that they are satisfactory. I understand that they can have some maybe this, maybe that, or some just so stories. I just don't really, I'm not convinced that they're empirical would be my point. And I know I went on a diatribe there, which means I just want to give you as much time as you want uninterrupted. I just wanted to just make my position as clear as possible. So go ahead.
Right. If I may, if it's no trouble for you, would you mind pulling up the nested hierarchy of vehicles you showed earlier?
Yes. Yeah. Just one sec. Okay. Here we go. Share screen.
By the way, just to let you know, we're at an hour and a half now, and I didn't know how far you want to go, basically.
Good question. Actually, time's flying by. Maybe we'll move on to this topic, and then because I know there's a couple after shows too, so I don't want us to hog the entire night. So we'll try and end it before the 2 hour mark. 2 hours is usually good for a discussion. So here's the, here, this one right here.
Yeah, that one. Now we can fit those into a nested hierarchy, I guess. My disagreement basically is the fact that we can fit designed objects into a nested hierarchy at all, because they do not have a history, or they have a limited history, I guess I should say. Now, what you can see, for instance, you can see that some traits, for instance, are correlated. Now, what you can find is, let's see, that, for instance, the what'd you call it? The steering wheel, for instance, in cars, for instance, and the turning apparatus in bicycles and unicycles, things like that. You can group them based on that. You could also group them based on the manufacturer, or, for instance, like I said, I don't know much about car anatomy here, but like you can group them based on a variety of traits, and what you get is several different trees or more that often conflict with one another.
Well, wait, stop. But here's the thing. Now we're going in a circle, because that's why I pointed to. I'm only stopping you just so we utilize the last half an hour the best we can. That's why I pointed to incomplete lineage sorting, where you've got all of these phylogenetic trees that are conflicting with each other, that are contradictory depending on what you're looking at. And fascinatingly enough, even when you look at certain types of RNA genetic sequences, now we're even finding incongruencies across a variety of different gene types or even DNA elements. And I've got tons of papers here. Are you familiar with the problem of incomplete lineage sorting, or?
Not exactly. I guess if I may bring up a point earlier, a point that I didn't that I forgot to mention, basically.
Yes, go ahead.
So you're referring, for instance, by the assumption of ancestry, basically to a time axis, basically, that this order of characters at various branching points, basically, right? You're talking about how we root the tree, essentially. Is that what you were referring to earlier by assumption of ancestry?
Are you talking about the overall assumption of the phylogenetic tree, or the assumption behind specifically incomplete lineage sorting?
Well, like what I was trying to get at was basically, for the most part, when you create a tree, you create it as unrooted, for instance.
Right.
So there's no you can look at it as basically showing relationships, but not necessarily universal ancestry. It's
Well,
only
I would just say that it's the relationships based on the sequences being used, and the trees, whether rooted or unrooted. These tree building programs give you the option, right? Do you want a rooted tree? Do you want an unrooted tree? It's the inference of relationship that is behind the ultimate assumption of common ancestry, because they're still assuming that whatever genetic sequences they're looking at, whatever traits they're looking at, they're still assuming that these all have been derived, right? Shared derived synapomorphies derived from previous common ancestors. But even quotes after quotes in paper after paper, as Speed of Sound loves me saying, talks about, because tree analysis tools are used so widely, they tend to introduce a bias into the interpretation of results. So that's what we need to focus on, is the interpretation of results, right? Is the interpretation consistent with the data, or is the interpretation of, say, the common design model more consistent with the data? So that's the thing, you know, all these phylogenetic trees, cladistics, cladograms, you know, yes, they demonstrate the hierarchy, but they don't necessarily demonstrate the relationship. And I see what you're saying, and it's a good point, and I want to see if I kind of understand what you're saying, if this answers your question. And then take your time after that, I'll yield.
Sure.
Because we look to this model of front-loaded genetic diversity, okay, or created heterozygosity, Adam and Eve, okay, according to the Bible, we're commanded to be fruitful and multiply. Did God want this to be carried out through cloning? Of course not. That wouldn't make any sense. So then it wouldn't make any sense for God to create Adam and Eve and the different kinds of animals genetically homogeneous. Essentially, they'd be reproducing clones, right? You'd want them to reproduce variety, okay? So God would have front-loaded Adam and Eve and all the created kinds with front-loaded DNA differences and variation-inducing elements that we would look to as endogenous retroviruses, ALUs, for example. So here's the thing. If we assume the created heterozygosity hypothesis, which I believe is the more plausible explanation because of the lack of evidence for abiogenesis, but that's a totally different subject. I just want to point that out because oftentimes people like Dr. Dan will say that hypothesis is post hoc ad hoc. Well, when abiogenesis is such a failure at this time, what's the most plausible alternative? And I would state that this hypothesis of front-loaded DNA capacity is a better hypothesis. Here's the point, then I'll yield. If scientists were to arrive at the creation event, let's say an hour after creation, okay, and they started doing analyses, and they started classifying animals based on morphology, anatomy, physiology. They started looking at genetic sequences. They started building these phylogenetic trees. They will conclude, okay, even though this was an hour after creation event, an hour after all these animals were front-loaded with genetic diversity, okay, they would still conclude nested hierarchies where, if they weren't aware of the fact that these animals were just created, they would then conclude some type of descent with modification, some type of derived ancestry, even though that creation event took place an hour before, right? It's going to reflect essentially from an evolutionary standpoint and evolutionary assumptions, even though it's an hour after creation, they will still conclude evolutionary history, even though there is no history. That's just the nature of the created heterozygosity hypothesis, that these genetic sequences that are front loaded, that code for the anatomy, the morphology, hierarchies are going to exist even at creation. Does that make sense? And just take as long as you want. I'd say I'd mute, but every time I mute, you go on robot mode. So
Yeah.
Go ahead.
So I guess what I'm kind of confused about, in terms of your model, is this created heterozygosity. Does it involve mutations, for instance? Like I'm kind of confused how it involves a pattern of history, but it itself does not include history. So could you clarify that?
Say that last little part again. I kind of looked at the chat real quick.
Right. Well, like you're saying that if we were to look at these kinds at creation, for instance,
Yeah.
And you're saying scientists would say a nested hierarchy. Just for clarification, like how many like roughly, like what kind of diversity are we talking about here? Like in terms of how many species or kinds there are, just so I can get a feel of what we're talking about.
That's actually a really good question. And you ask good questions that require detailed answers, but I'm going to make this as short as possible. So the best way then for us to determine ancestry and determine how much variety or diversity was present at the creation event, it's probably best to look at the global flood event, because that would have been the bottleneck. Okay, and that's why I've got diagrams here of the arc event where we know, let's say essentially you've got two cats, okay, that were initially front loaded with all these DNA differences, DNA elements, okay, at the flood. Okay, they would have lost a little bit of that original created heterozygosity, but by definition, the flood bottleneck was one generation followed by rapid growth, rapid population growth. Okay, so only very limited of the diversity would have been lost, and that's why we have study after study, and I've got a ton of slides on it. I won't go into it right now for sake of time. There's been studies by the secularists on genetic diversity in highly conserved mitochondrial genes, for example, the CO1 gene, that demonstrated that over 90% of life, and I'm sure you've heard me say it before, has come into existence at the same time, which we would not say is the creation event. We would say this is the flood event, because essentially all this mitochondrial DNA diversity was reset, okay, due to that bottleneck. So now we can look to mutation rates and we can determine ancestry that way. When we go to the creation event, a couple thousand years before, God tells us he created Adam and Eve, okay? That's two people. That would restrict diversity. So therefore, if we're doing tests today in the present, we would then assume or expect or predict that humans overall would have low genetic diversity. God created populations of animals. Okay, it wasn't like the ark event where he brought 2 of some, 7 of some. He created populations of animals in fully functioning ecosystems. Okay, so that would then give us the expectation that animals would have a higher genetic diversity because they would have started in populations. And that's what we see when we sequence animals. For the most part, we see they have a lot of genetic diversity. Chimpanzees, within chimpanzees, there are varieties of chimpanzees that have more diversity within themselves than humans and Neanderthals have. That's how much diversity animals have, which makes sense because it looks like God gave animals the greater ability to diversify. He gave them greater levels of heterozygosity versus humans. So to answer your question, it's best to look at the ark event. But if you want to go back to the creation event, the best answer I could give you is humans overall would have low genetic diversity. Animals would have higher genetic diversity. Does that make sense? Does that kind of answer your question?
Yeah, what I'm trying to figure out is, like, for instance, say we have trying to think through this. Basically you have, let's just say a cat kind. So you have the basic form that would diversify. and you have
Oh, I think we kind of lost you there for a second.
no.
Your audio just went into.
Sorry, I'm thinking of how to try and word this.
Okay. Because you did kind of you went robotic mode for a second and then silent. So, but no,
okay.
go ahead. A couple of people in the chat say we lost audio, but no, we're good. Go ahead, take your time.
I like to think things through basically. So essentially, this kind of gets into the problem of reconstructing the ancestral states of the original created kinds, because without knowing what those states were, for instance, I guess what I'm trying to say is, without knowing what the ancestral states were, it is very difficult to say whether or not we would still find a nested hierarchy, because it depends on what kind of what types of features they had originally. So for instance, this kind of gets back to the how do we differentiate between universal common, and I notice it isn't exactly what the debate topic is, but how do we differentiate between universal common descent and these created kinds, basically? I guess answer that while I see if I can if I have anything else to add, basically.
Yeah, no problem. But I also want you to feel out of fairness that you're getting as much time as you need, equal time to respond to everything. I'm happy to answer that question. Is there anything else you wanted to add to that before I do?
No. Yeah. Go ahead.
Okay. So I'll put it into a nutshell, because there's a variety of ways that we've gone about identifying what's related, what's not. Okay. Function and mutation rates, I would say, are key. Mutation rates primarily in the uniparentally inherited DNA, okay? The Y chromosome, which is passed down through fathers, through males, and mitochondrial DNA, which is passed down from our mothers, from the woman's side, okay? Those differences in those DNA compartments, we can agree with the evolutionists, are the result of mutations, okay? Therefore, we can look at the number of DNA differences in those DNA compartments and make predictions, retrodictions, and see how far back can we go. And that's why I oftentimes look to these mitochondrial DNA phylogenetic trees. You got to go all the way down here for it. Y chromosome phylogenetic trees that show overall, this is humans. We can do the same thing with animals. Dr. Nathaniel Jensen's making a ton of predictions on different types of animals. But whether you look at the mitochondrial genetic phylogenetic tree or the Y chromosome phylogenetic tree, what you see is a pattern that reflects the biblical model. You also see different branch lengths, even in certain haplogroups, like this one, HVR in Europe, most common European haplogroup. You see different branch lengths, breaking the assumption that the evolutionists make on molecular clocks, because people, okay, are picking up more mutations than some of their cousins, and they go back to the same ancestor. Okay, so if you can pick up more mutations in the same amount of time, there's no real way that the evolutionists can now make a prediction or make an assumption as to when chimps and humans split and then get a mutation rate from that. So point is, we see only a few mutations in mitochondrial DNA. Okay, a few mutations removed from the Eve sequence. We have the Eve sequence. Same thing with the Y chromosome. Only a few hundred generations worth of mutations in the Y chromosome, a few hundred mutations in the mitochondrial, a few hundred generations worth of mutations in the mitochondrial DNA. That's just mutation rates. We can go into DNA function, so we can look to whether or not the pseudogenes, where I've got a ton of papers here on pseudogenes, whether or not they're functional, right? More consistent with the design hypothesis, or are we looking at a lot of genomic fossils? Are we looking at a lot of evolutionary leftovers? And the data seems to suggest, I mean, here's 4 papers alone that go over all these retroviral elements that evolutionists assumed were non-functional. We now see that they populate the genome in functional ways, functional purposes. So, obviously we can go into probably 10 hours worth of conversations on this, but taxonomically restricted genes would help us determine which creatures are related and which creatures are not related. So in a nutshell, that would be my answer. DNA function, DNA elements, okay, like ERVs, retroviruses, and we can look to DNA barcoding, of course, as I talked about earlier with the CO1 gene, and mutation rates. I think mutation rates are huge. That's probably the best answer I can give you, but we can look to, so I know you saw, we can build our own phylogenies. So I've got phylogenies here. So we got one of humans, just based on DNA function, based on mutation rates. Okay, we've able to determine that humans, Y chromosome dissimilarity between chimpanzee and human, it's less than 35% when you consider overall architecture, gene content. That's a huge, huge difference for the evolutionists to explain. So here we got a phylogeny where you start with humans, okay, not related to any of your other Australopithecines or your chimp-like ancestors, gorilla-like ancestors. No, we've been able to build a phylogeny where we've got humans at creation, right? We've got some side branches: Arcticus, Neanderthalensis, Denisovans, Naledi, Floresiensis, Luzonensis I'd put on here as well. These are extinct groups, either who migrated a little bit before Babel or after Babel, okay, which resulted in inbreeding and accelerated mutation accumulation and essentially extinction. We got humans today. So we've been able to separate humans from the rest of the biological world through these numbers of lines of evidence that I just went over. And you ask great questions that require detailed answers, but that is my answer in a nutshell. And what I'm going to do, because you're my guest, I'll give you the last word on that, okay? Give any rebuttal to any of that you'd like to, and then kind of go into a wrap up, conclude your statements, give some thoughts on how you felt this went, and then we'll kind of start wrapping it up there. Andrew, go ahead.
Yeah. So I guess to make my final point, would it be okay if you pulled up the last image that you had on screen?
Yes, just one sec.
Or
Actually, Andrew, give me 1 sec. I got to run upstairs real quick. Just give a couple rebuttals real quick, or say whatever you'd like. I'll be back in about 1 minute.
okay. Oh, I don't know if I'm roboting. So I guess one thing I would say. Oh, I feel like I might be roboting. Am I still roboting? I feel like I am. Let's see. What is my favorite? Oh, what is my favorite flavor of ice cream? Chocolate, obviously.
All right. See, the problem is when my wife works midnights and one of the kids Like my daughter, she's 4.5. The second she's out of water, all I can hear is banging upstairs. And since I'm the only one home, I got to go upstairs and refill her water bottle. So, but I can still hear because I got my phone here. So if you said what you feel needs to be said, what I'll do is share screen. I know you wanted to.
Yeah. I think I might have been roboting. The chat was saying I might have been roboting.
You probably were because I muted myself. So apparently, have you ever had that where if someone else mutes, all of a sudden you're just in full robot mode?
No, not really. Can you go back to the one of the human ancestry?
Oh yeah.
The orchard, basically. I guess the created orchard.
Right there.
I guess some people call it. There we go. So I guess my final point would be that we see these similarities, these synapomorphies, for instance, between gorillas, chimpanzees, orangutans, what I would call transitional humans, and Homo sapiens, of course. However, when we look for like the loss of tail in apes, just to give an example. Now, when we look at monkeys, for example, when we look at an outgroup, we can see that those synapomorphies are not present because, of course, most monkeys. I believe there are some exceptions, but most old world and New World monkeys have tails. They do not have the same kinds of traits as apes, for instance. I'm not a primatologist, but I guess to kind of wrap up, I feel as though this pattern reflects a kind of shared history. Basically, you would expect this if there were a history. You would not expect this if this was a pattern of an orchard, for instance, that started at one event but did not start at a common ancestor, essentially. So I guess that would be my main point from this discussion.
Well, if I could ask then, because I believe ultimately, okay, this comes down to the question of ancestry, which then as a result comes down to how we view and how we interpret the evidence, okay? So when it comes to the anatomy, the morphology, when it comes to shared derived synapomorphies, okay, these shared derived traits essentially that are assumed to have been derived from past common ancestors, I want to emphasize the assumption aspect of that. But here's the thing. If we're both predicting patterns, hierarchical patterns, we're both predicting homologous patterns, we're both predicting mosaics, okay, we can also pick at each other's model out for hours and hours worth and say, well, what about this? What about this? But overall, the rule is a hierarchy. The rule is homology. That's why when we go to the differentiating lines of evidence, because what you were saying there still seem to be assuming the shared derived synapomorphies have actually been derived, and that maybe God created, an old world monkey kind, a great ape kind. I would essentially, based on the genetic data, especially the Y chromosome, it looks like God originally created, obviously, humans, a gorilla type, a chimpanzee and bonobo type, where your chimpanzee and bonobo goes back to a common ancestor, and an orangutan type. Because the Y chromosome, for example, in humans, when you consider overall gene content architecture, is actually more similar between humans and gorillas than it is between humans and chimps. And based on this incomplete lineage sorting, okay, depending on what gene they look at, they're actually getting these trees where humans and chimpanzees are not the closest to each other. They're getting trees where gorillas and humans are closer, or humans and bonobos are closer, right? So you got all these contradictions. But if you still want to assume ancestry, as I feel like you're doing, when it comes to the differentiating lines of evidence. And it's not like I'm not trying to trap you or anything. I am just curious because this is one I commonly bring up. And I'm just curious, because I find the data with the Y chromosome so fascinating because, for one, the evolutionists didn't predict it. Two, all I've seen is hypotheses in the form of explanations that haven't really been tested. Maybe this, maybe that. Maybe the sexual nature of chimpanzees led to a faster mutation rate, and maybe chimpanzees are the odd man out, versus humans and gorillas. These types of hypotheses. How do you explain essentially then? Why is the chimpanzee and human Y chromosome? As you can see here, this is the paper from Secular Journal. Chimpanzee human Y chromosomes are remarkably divergent in structure and gene content. So to get that amount of change, less than 35% dissimilar with the Y chromosome. Remember, the Y chromosome is non-recombining DNA, so essentially it should be more stable. How do you explain that incredible dissimilarity from your view that you're defending tonight, the hierarchies by descent model? Empiricist, go ahead.
So again, I'm not an expert on primatology, for instance, so I'm probably just spewing out nonsense at this point. But I think there might be some evidence that the Y chromosome or some other similar chromosome evolves more rapidly, I believe. Again, I'm probably I would just have to say I don't know, and I'd have to look into it, unfortunately.
Oh, and that's completely fine. I wasn't trying. It's just because, and I tried to keep this conversation more in line with, hey, let's just focus on the hierarchies. Let's focus on the cladistics. But there's just so many differentiating lines of evidence that I've never seen a good answer to. And the answer you just gave is one of the answers that's provided is a rapidly evolving Y chromosome. Well, let me ask you this then. Okay, here's the problem with that. Every single male Y chromosome on this planet is nearly identical, okay? There's incredibly low genetic variation in human male Y chromosome, okay? This is a Y chromosome phylogenetic tree, Simon's Genome Diversity Project, okay? Reflection of DNA differences. This is unrooted in order to give us a natural reading of the diagram. If evolutionists are saying that this Y chromosome goes back hundreds of thousands of years, but yet it's rapidly mutating, like you just said, as a possible explanation, then how in the world have human males managed to keep a Y chromosome that's literally 99.9% similar worldwide? Because we know there's people groups that migrate, they diverge, right? Because we've got geographically specific Y chromosomes. Why don't we see any highly mutated Y chromosomes, highly divergent Y chromosomes in people groups around the world? How, in a rapidly mutating Y chromosome that's apparently old and goes back hundreds of thousands of years, how has it been able to maintain such low genetic variation would be my question.
Right. So I guess just to clarify something, so is this male Y chromosome, is this based on a specific population, or is it based on all humans, or like a certain continent?
That's a very good question. No, these are all people groups. Africans, Europeans, all people groups.
Okay. Yeah, my best I guess I could say hypothesis at this point, because maybe you could test it, would be potentially that there was well, I know that there's a specific bottleneck event that we would say is 75 or so thousand years ago, due to probably the Mount Toba eruption. So potentially that's it. Some kind of bottleneck event, basically, would be my best guess.
And good response. So a couple things. The evolutionary community, I document this in my book, they've essentially invoked many bottlenecks. One 200,000 years ago, one possibly with Erectus. Okay, you've got Erectus for millions of years evolving. Erectus essentially inhabiting a lot of the planet. One question I might have to save it for another discussion, but if we've had essentially 12 billion Homo erectus bodies on the planet that have been born and died, who even the evolutionists would agree knew how to create fire. They had civilizations, communities, they had navigational skills. 12 billion bodies that we should find in the ground, and yet if you take all the erectus bones or erectus fossils, you can literally fit them in the trunk of a car. So there's no evidence for this million years worth of Homo erectus being born and dying. We should have evidence for these 12 billion bodies. But whether you look to a bottleneck with erectus, or say 200,000 years ago, or 70,000 years ago with the toba extinction, what I find funny about the toba extinction is a lot of evolutionists now are actually avoiding the toba extinction argument. I've heard it even from evolutionists in this community because there's so much conflicting data that, for sake of time, I won't get into. But even let's just, for the benefit of the doubt, let's even say 70,000 years ago. For one, the next question would be, how is that bottleneck even remotely feasible? Because 70,000 years ago, you got this massive bottleneck, which you're saying and invoking in order to reduce genetic diversity in nuclear DNA, Y chromosome, and mitochondrial DNA, right? Because if we're evolving for millions and millions of years, we're generating more and more mutations every generation, which means we're generating more and more diversity, which means humans should have high genetic diversity all around. We have low diversity, therefore maybe an extinction event wiped it all out, reset the Y chromosome clock, reset the mitochondrial DNA clock. Now here's the problem. The evolutionary community cannot invoke a one-generation bottleneck followed by rapid population growth like us. No, because that wouldn't be enough time to reduce genetic diversity. So the evolutionary community has to invoke what's called an extended population bottleneck, which means you've got roughly 2 to 10,000 people, okay, who have been subject to this incredibly damaging near extinction event in this out of Africa scenario, where now you've got all of these previously accumulated mutations coming to the forefront. They're manifested, okay, leading to rapid and accelerated genetic degeneration, because that's what happens with inbreeding, is these recessive mutations come to the forefront, right? So that would be incredibly damaging. But then all of a sudden, this group of humans explode into all parts of the world, seizing dominion over the planet after a nearly damaging extinction event like this. I mean, we can see an echo of this in the cheetah population today. They've been under a population bottleneck. They're down to like 7,000. Their sperm's degenerate. Conservationists are worried about their extinction. And we're not going to see anytime soon these cheetahs just exploding in all parts of the world, seizing dominion over the planet. I know that's kind of tongue in cheek, but that's kind of what the evolutionists are invoking with the population bottleneck. But I'll yield after this. For sake of argument, let's just say the toba extinction 70,000 years ago. Even 70,000 years worth of a rapid mutating Y chromosome, especially in the out of Africa scenario, you get all these different people groups going to different parts of the world, and now they're isolated and separated from each other for thousands of years. What that means is you should have these geographically specific Y chromosomes in different parts of the world where these people groups are isolated from each other. We should then see, we should see Y chromosomes that are highly divergent from each other around the globe, highly mutated. Right? With our model, we only invoke 4,500 years since the bottleneck. So 4,500 years of a rapidly mutating Y chromosome, that makes sense. It's only been a few thousand years. Therefore, we would expect a Y chromosome worldwide to be incredibly similar with low genetic variation. That makes sense. But even your toba extinction 70,000 years ago, we'd still expect a Y chromosome, some Y chromosomes that are highly divergent. So I do appreciate you giving that answer, because I have, if you got that at the top of your head as a hypothesis, that is a hypothesis that's been invoked, as well as a number of other hypotheses. But every hypothesis that I see invoked comes with a massive number of problems associated with it. I know I spoke a lot there because there was a lot to talk about with your answer. So once again, since now we're going over 2 hours, why don't we give you the last word on all of this and respond to anything that I just said, if you'd like to, of course. I know I don't like to leave anything hanging. Therefore, you can feel free to do the same. And I will say this has been a great discussion, a respectful discussion. I'm happy to do this any number of times again in the future. So yeah, I'm going to hand it over to you. Respond to whatever you want to, and have some final words there.
Yeah, I would, I would say that, obviously I might have my bias towards universal common descent. I feel like the pattern that we see in nature reflects it better. I guess now some of the things you mentioned, of course, throughout this discussion, actually, unfortunately, I've been unable to respond to, partly my brain is fizzling out, but also partly because I'm not an expert. But I feel that definitely, we could definitely do another discussion on this, or something on mutations and genetics, for instance. I do like that subject, or even maybe like astronomy, for instance. Those are kind of my main subjects. But yeah, it's been a fun discussion so far, and I appreciate letting this remain very cordial.
Awesome. I agree. And I'm glad we could fix the audio problem, because the second you pulled up your slides, I was thinking, these are some of my favorite topics, getting into cladistics, taxonomy, nested hierarchies, homologous patterns. So even though I had to stay unmuted the whole time, I think overall we fixed the problem, and it's been a good discussion. It's been cordial. Like I said, I'm going to give you the last word there, so I won't make any other comments in terms of the discussion. And I appreciate you making this such a good discussion on a lot of heavy topics. I hope the audience enjoyed. And it looks like our good friend Speed of Sound has an after show right after this. So everybody head over to good old Speed of Sound's channel. Thank you for everybody in the chat. I hope you enjoyed this. And lots more discussions, debates, interviews to come in the future. So once again, I can't believe it's been over 2 hours. It's flown by. So Thanks for being here, a rational empiricist, and SFT's out.